So good afternoon. I wanted to welcome everybody to the February edition of the Maternal and Child Health Research Institute seminar series, where we're really focusing and highlighting some of the innovative and amazing programs we have at MCHRI. And today is really one of the outstanding programs at Stanford with Kara Davis, who will talk to us about her studies on single-cell analysis of leukemia heterogeneity. But first, we wanted to get to some housekeeping issues. Next slide. So really wanted to point out this amazing lineup coming up of the next few MCHRI seminars. This is all available on the web, but March 1st next month, Michelle Munch and Juliet Mills are going to tell us to you about maladaptive myelination in pediatric epilepsy. In April, Gary Darmstadt will talk to you about gender inequalities in community health globally. Again, an amazing project with the Gates Foundation. In May, Julie Simmer on maternal health topics. And here and you see the rest of these, really an amazing lineup of throughout the rest of the year. These are all available on the MCHRI website. Next slide. And then also wanted to point out that Thursday, October 28th, is our 4th annual MCHRI Research Institute Symposium, where we highlight really 3 emerging topics in the Stanford maternal and child health community. So please, and then also a great abstract and community development. Right now, it's scheduled to be hybrid, both in person and virtual, but that may change as the pandemic changes as well. So please keep that date available, Thursday, October 28th. Next slide, and then this is our education committee. Please contact any of us for suggestions or improvements, or if you want to join, please contact me. Next slide. And today, after this talk, we're going to send you an online survey form for any suggestions about the talk or how to make it better. So please respond to the survey. And today, we wanted to introduce Kara Davis, who's an assistant professor of pediatrics of Hem/Onc at Stanford. We're excited to have Kara present today because she is a recipient of the MCHRI pilot grant from 2012 and a faculty scholar award in 2019. She was a Hem/Onc fellow here at Stanford and really has now translated her fellowship into a really innovative maternal and child health and particular pediatric oncology translational biology, which she'll tell you about. I really think she really highlights the uses of single cell analysis and heterogeneity, as well as this amazing Hem/Onc sort of powerhouse that she'll tell you about with for pediatric cancers. And so I'll have her now start talking, but also there's a chat, question and answer function on the website. So please put your questions there, and then afterwards we'll have a Q&A session. So Kara, thanks for joining us and look forward to your talk.
Thanks, Tony. Thanks so much for the invitation to be here. Let me get my screen properly shared. Hopefully that looks good for everybody.
That looks great.
Great. Yeah, thank you for the opportunity to put on lipstick and not be speaking with a mask on. It's kind of exciting. So we really, in my lab, are interested in not just leukemia, which is mostly what I'm going to focus on today, but childhood cancer. But we are going to talk about how we approach these childhood cancers and use B-cell acute lymphoblastic leukemia as our example today. It's the most common cancer of childhood. And in the 1960s, it was pretty much routinely a deadly cancer of childhood. So only about 20% of patients survived. But it really has become a poster child for success in cancer treatment, and I would say across the board through cooperative clinical trials and successive clinical trials. And this Kaplan-Meier curve is from St. Jude, looking at their successive clinical trials to now, and this is even dated 2007. But we can say to parents who come in with their kids with B-cell ALL that greater than 90% of them are likely to be long-term survivors, which is really wonderful. However, we still deal with the fact that relapse leukemia remains a major problem and is the #2 killer of children with cancer, just having been surpassed by brain tumors in the last couple of years. On the left side of the slide here, I have some Kaplan-Meier curves from studies that were done looking at relapse leukemia survival from 1988 to 2002. And what this really highlights is in that era, at the early side and the late side, we didn't really see any improvements in our approach to relapse leukemia. So we haven't made a lot of improvement there. And then on the right side, this Kaplan-Meier curve is from the clinical trial in which led to the approval of CD19 CAR T cells for childhood leukemia. We were a center that ran that trial. And as we could see, in fact, we could do better as far as getting patients out. This is not quite 2 years, but we still had a significant amount of patients, if we look at the event-free survival curve, that were relapsing and did not get durable remissions in their relapse. So relapse is still a really big problem, even though we've made great strides for the disease. And so really the goal of what we do in the lab is to understand what cells actually are responsible for causing relapse. So at the time of a patient's diagnosis, they have billions of cancer cells. And clinically, what happens is they're diagnosed, we treat them with chemotherapy. And over the course of the first month or so, we assess what we call minimal residual disease to try to see how much leukemia is left, hopefully none. And then they go on to finish their therapy, and the majority of patients will be cured, but we're really, sort of trying to figure out this 20%, 15 to 20% of patients who won't be cured. And so we want to know, can we identify in the diagnostic sample predictors of relapse or predictors of minimal residual disease? And then can we understand what those cells are so that we could actually prevent the relapse from happening altogether? And so the main tool of our trade and of our armamentarium is mass cytometry, which I describe, and I think most people at Stanford know about because it's sort of grown up here, and we have many, many mass cytometers on our campus, luckily. But the way our standard experimental procedures work is that we will stimulate cells in vitro, do a fixation protocol, stain the cells with antibodies that are tagged with isotope labeled antibodies. So similar to flow cytometry, except it's not a fluorophore that we're looking at. And then as the cells go through the instrument, they're measured by a time of flight mass spec on the single cell level. And the data comes out looking very much like what you would expect from a flow cytometry plot. So to give some background, and this is on a published study that sort of lays the foundation for how we are now proceeding. We had looked at a cohort of 60 patients with ALL in their diagnostic sample and some healthy control bone marrows. Subjected cells to ex vivo stimulation as a way to reveal signaling patterns and relevant patterns to their developmental state. We stained the cells with a panel of antibodies that encompassed about 40 proteins that are both surface markers and phenotype proteins, as well as transcription factors and signaling molecules to help us understand a little bit about how the cells are wired. And then, of course, we looked at them on the CyTOF. And before we started this particular study, when we did initial studies with the CyTOF and we started to do sort of clustering analysis, this is of 9 bone marrow patients that we had from St. Jude. Here, every one of these clusters is colored by the patient that it came from. And we could really see that every single patient became an island, and we weren't really finding sort of shared phenotypes by taking this approach from an analysis standpoint. So with our larger cohort, we turned to our history or our prior knowledge of normal B cell development as a way to align these leukemia cells. And just to say, when I was a postdoc in Gary Nolan's lab, I got the privilege to work with Sean Bendel and Alada Mir on a paper that we looked at healthy human B cell development using the CyTOF, and we're able to really outline what is the trajectory of normal B cell development with this sort of single cell approach. And so when we then turn to our leukemia data here in the middle plot, if we project the healthy cell populations that we discovered in our early work into PCA space, they can almost make a straight line through them. I hope you guys are seeing my cursor. And then if we took, for example, for leukemia samples and put them into the same PCA space, we could see that not only did they share space with normal progenitor populations, they also shared space with each other. So we thought that this was an interesting approach, and we took it further by actually developing something a little more fine-grained than what we were getting with PCA, and we developed a single cell developmental classifier. So what we did was we took all of our healthy control populations and defined them based on what we knew was the developmental trajectory. Each one of those populations was essentially given a barcode that defined its developmental state based on the expression of 11 proteins, which I'm showing here zoomed in. And then for each of those 60 patients, and we had on average about 600,000 cells per patient, we would match them to their most similar healthy population, therefore giving them a developmental identity. To speed this up, we then used that data in combination with an elastic net model to try to see if we could find any of those populations that were associated with future relapse. And we called this model the developmentally dependent predictor of relapse, DDPR, or we call it DEEPR. And so here I'm showing you that with this model, we found 6 features that were present in the diagnostic sample that we could then associate or were highly associated with future relapse. And these these features, to summarize them, were all signaling features, and they were actually all in 2 cell populations that are developmentally right next to each other. So the transition from a late pro-B cell to an early pre-B cell. And the interpretation of these is essentially that patients who were going to relapse had baseline activation of these signals that could not be further induced. And that's what sort of made them stand out compared to patients who were not going to relapse. So we looked in our bigger cohort now at how did these predictive features sort of play with the clinical metrics that we use to predict relapse risk. So on the top left here, I'm showing you the Kaplan-Meier for the NCI-Rome criteria, which is the very standard, tried and true metric of relapse risk prediction. And if we only use that metric in our cohort, we see a nice separation between patients who are standard and high risk. If we go down now to the bottom left and we add our deeper risk metric, or as a low or high risk patient, we actually can further stratify those Kaplan-Meier curves such that we can find patients who were standard risk but may have actually been at risk of relapse, and also patients who are at high risk but may have actually been in the deeper approximation low risk of relapse. On the right-hand side, I'm showing you the same thing using minimal residual disease risk, which is another now extremely tried risk classification system where we know that if we have small amounts of disease left after the first month of therapy, that those patients are at a higher risk of relapse. And again, the MRD risk, when we use it alone, has a good stratification of the cohort, but when we add our deeper risk score, we actually can do better. So we were really excited about these results. And, this is published a few years ago now, in which, we found these cells within the billions of cells at diagnosis that are characterized by these activated signaling networks, and that they're particularly in a particular developmental state. And we think that those cells are able to survive treatment and potentially come back and then mediate relapse. But the most exciting part of this was that it gave us a place to look or focus on, because of course, now we could get inside an individual, tumor sample and try to say, okay, now we need to, these are the cells we need to more quickly focus on to understand, how they cause resistance. And just to highlight, this work was, led by Zena Good, who is now a postdoc in Crystal Makel's lab, and Sylvia Plurvitis' lab, Estrella Yeager, who's still with us here, and Yolanda Sarno, who's now a postdoc in my lab. So I'm going to now transition. I want to try to talk about two stories that are in progress or in process. They're not totally done, but some extensions of how we've used these tools in other facets. And this one, which is being led by Yuxuan Liu in the lab, is a direct extension of the work that I just went through. So as I mentioned, in that previous work, we had identified these active signaling cells as sort of the potential bad guys in relapse. And here, if I take two of those patients that we looked at and share their matched diagnosis and relapse samples in these early pre-B cells, we could see that in those signaling nodes that we had identified, that there is this coordinated signaling that changes or enhances almost at the time of relapse. And I have it circled here in this one patient where S6, CREB, and SIC are all actively signaling together in these cells. And we actually did a sort of more sophisticated analysis using an algorithm called Dreamy to look at what's called conditional dependencies. And we could find actually that at the time of relapse, the relationships between these three molecules actually strengthened. And so you could hypothesize then that there's a couple ways that could happen. Either you're breeding some sort of resistance and maybe a new mutation that may arise at relapse that is more dependent or influence the signaling relationship. You also could hypothesize that it's because the cells became less heterogeneous at relapse, meaning that at diagnosis, there's a lot of cells there that don't rely on the signaling network, but because these cells are the ones that then can initiate relapse, that at the time of relapse, there are more of them, they're more homogeneous population, so their relationship appears strengthened. And so to follow up on this particular network, we went back and thought a lot about what happens in normal B cells or pre-B cells as they're developing. And this is a really important developmental checkpoint as B cells sort of grow up normally. This is exactly the time in a B cell's life that it's rearranging its heavy and light chains to make an immunoglobulin receptor. And so what cells do at this time is they have what they call a pre-B cell receptor, which is how they sort of try on their immunoglobulin rearrangements to see if they work. And the pre-B cell receptor actually does signal in a similar pathway as to a mature B cell receptor, which we know is really important for healthy B cell survival and has been implicated in more mature B cell lymphomas like diffuse large B cell lymphoma. And it involves multiple pathways with downstream pathways, including PA3 kinase, as well as the RAS and ERK pathway. And just to point out here that SIC is very membrane proximal here. It's coming right, from that pre-B cell receptor. And previous groups, Marcus Muchen's group as one, have sort of classified ALLs based on gene expression and some components of the pre-B cell receptor into pre-BCR positive ALL, which is the vast majority of ALLs, and pre-BCR negative, which is the majority, and pre-BCR positive, and divided these up based on particular translocations or genetic rearrangements that are common in ALL. And so now with our prior data, we started to ask the question, is it not just potentially being a pre-BCR positive ALL, but is it that you're a pre-BCR positive ALL cell that could be really one of the things that contributes or is responsible for relapse? So Yuxuan first started by taking NOM6, a cell line that we have in the lab. It's kind of a workhorse for us, which is classified as a pre-BCR positive ALL. And she used CRISPR to knock down SIC or knock out SIC. And here in this curve, what we found was that those SIC negative cells were slower growing than the wild type and actually had a growth disadvantage when we had them, cultured in a mixed population, suggesting that the loss of SIC was detrimental to survival of the cells that had undergone the knockout. When Yuxuan looked at this now on the CyTOF by looking at a really sort of comprehensive signaling panel and compared the cell populations in 3 different knockdown lines, 2 different clones that had between, I think, about 50 and 80% knockdown, and then a mixed population of cells, we could see that with the sick knockdown, both in the baseline state and after the cells had been subjected to B-cell receptor cross-linking, that there was you had less activation of phosphosic, phosphoblink, phospho-BTK, all downstream of the pre-B-cell receptor, and as well as PI3 kinase and signaling in some of the pathways that came up in our relapse predictor, including 4-ABP1. And when she looked actually then at just the single cell clones, but not the mixed population, we also saw differences in S6 and ERK as well. So really suggesting that SIC was acting downstream of the pre-B-cell receptor. And finally, one other sort of interesting observation was that PAX5 seemed to also be influenced in these knockdown cells. And that was surprising to us because PAX5 is a really important transcription factor of B-cell identity. And mutations in PAX5 are associated with relapse in ALL. And so conclusions from this part in the NOM6 cells, we could see that SIC appears to be upstream of the activation of phosphocreb, S6, and other signaling molecules, and also appears to have some relationship with PAX5. And so, you know, like I said, this is not a complete story. So we're continuing this now by repeating the knockdown in other both pre-BCR positive cell lines or cells that are sort of classified that way, as well as in pre-BCR negative cell lines to see how does this look when we have these cells that don't appear to have this dependence on the pre-B-cell receptor. So Yuxuan also went and looked at public data sets. And so here is some data from CBio portal looking at the target data set, which is a big set of 200 samples. Some of these are actually diagnosis relapse samples. And we here specifically looking at the relationship between PAX5 and SIC, we could see a pretty decent correlation between those two in these in the data from this public repository. And again, this is sort of enriched for relapse samples. And in addition, along with Dora Jadwi, who is an undergrad who's been working with us in the lab for a couple of years, we also looked at the cancer dependency portal, which is out of the Broad, where they've done pretty extensive CRISPR screens in a lot of different cell lines. And here again, highlighted in the red are B-cell ALL cell lines, showing the effect of PAX5 knockdown and SIC knockdown. And here's NOM6 all the way down in the bottom left corner. So again, suggesting dependency of those proteins on for these cells. And the same can be done also with in the same portal, looking at some proteomics data. So together, then we went and back to look at our 60 patients and see what is the relationship between SIC and PAX5 in those patients. And although in that data set, we didn't have total SIC as a measurement, we were measuring activated SIC, so phosphorylated SIC. And all the way on the left, I'm showing you, if we look at all of the diagnosis samples, and these are the patients who are not going to relapse, that there's a very poor correlation between phospho-SIC and PAX5. If we look at the diagnostic samples for the patients that we know are going to relapse, we see a strengthening of this correlation. And then if we specifically look at the relapse samples themselves, the correlation becomes quite striking at 0.97. So, and this is again in the bulk patient cells, not even in our specific cell population. So this really does look like SIC is upstream of CREB and S6 and may regulate PAX5. One of the things now we're investigating is, the role of total SIC versus activated or sort of phospho-SIC downstream of the pre-BCR, and whether or not it's possible that there are post-translational modifications of PAX5 that are regulated through the signaling of the pre-B-cell receptor, as this has been shown in multiple myeloma there's a phosphorylation event that happens to PAX5 downstream of the B-cell receptor. So those experiments are ongoing, and we're excited to learn more about how these things are related. And so just to sort of start to build this model, as I mentioned, we're trying to understand this relationship between the sick and PAX5. So Yushuan also at the same time was looking at, generated some RNA-seq data from our sick knockout cells. And here we're just showing the clustering between the two different cell states. So the knockout compared to the wild type, and the volcano plot to show the significant genes, upregulated and downregulated genes as an overview. But when we started to look at GSEA analysis, what we found was that highlighting that there were changes in cellular metabolism in the sick knockout cells that we did not expect, and also I will say Mchri targets. But some of the top pathways that came up are OXPHOS and then mTORC signaling, which is not surprising based on our previous CyTOF data. Here's a list getting into the OXPHOS pathway, and in particular, looking at a lot of proteins in this pathway that are related to mitochondrial proteins, which has previously been published by just actually last year by John Dick's lab using a set of patient-derived xenografts, also implicating potential changes in cellular metabolism in relapse. We also then went back and looked at some RNA sequencing data that we generated from patients that had been profiled for relapse on the CyTOF. So in our what we call deeper high patients, we had 3, and deeper low patients, we had 3. So these are diagnostic samples. We sorted them for this pro-B, pre-B population and sent them for RNA sequencing. Here again is the clustering of those two patient sets, and we can actually see that the patients who are high risk for relapse clustered together with the patients with low risk clustered together. And interestingly, if we look specifically at the gene pathways that are different in the high risk for relapse patients, again, we come up with the top hits being Mchri targets, oxidative phosphorylation, and mTORC signaling, among others. So, we're starting to see this, this metabolism and signaling relationships that certainly we suspected the signaling relationships, but now coming up both in our cell lines with the knockout and then also in the actual primary patient samples where we already had developed this model. And again, here's just a different way of showing the GSEA enrichments, where we can see again that these pathways are quite enriched in the patients who are in the cells from the patients who are going to relapse. Going back to the DeepMap portal, again, Dora took a look then at the data from pre-BCR positive and pre-BCR negative cell lines. And if you look specifically in the pre-BCR positive cell lines and the genes that are upregulated there, that again, we see pathways related to mitochondrial translation, the TCA cycle, electric transport chain. So pointing all in the same direction as the data that we've been generating in the lab. Yushuan then has been learning, and we've been learning together about cellular metabolism and how to look at that. And so doing some fairly simple experiments we've been able to do during the, sort of COVID restrictions, looking at mitochondrial content in relationship to total sick in the cell lines that we have in the lab. We didn't see a strict relationship between total sick and mitochondrial content as a function of the pre-BCR category of these different cell lines. Although interestingly, we did see, for instance, NOM6 cells, which, like I said, are our workhorse, are both high for mitochondrial content and sick. Then when she took a look at the ratio between active mitochondria versus just mitochondrial content, we could see that the knockout cells appear to have more active mitochondria compared to the wild type cells. And doing some further experiments looking at reactive oxygen species, the sick knockout cells also appear to have higher levels of ROS. So we're following these up, and we're learning actually how to use the seahorse and to try to get more sort of functional readouts of these different cells, which then we would extend to other cell lines, as we mentioned, based on their BCR status. But one other tool that now we've been integrating, and this is based on this great paper that Felix Hartman published out of Sean Bendel's lab this past year, where they basically developed a CyTOF panel to look at metabolic features of cells to be able to do what they call single cell metabolic profiling. Graciously, Felix gave us, some tests worth of this panel. So we were able to look at them on our sick knockout cells. Here we found, actually, here I'm showing on the wild type versus the knockout, interestingly, that if we gate on sick content, that we actually, in the knockout cells, we're still getting some signal from our phosphosick antibody. We're hypothesizing that actually may be some cross-reactivity with phospho-ZAP70. ZAP70 is sort of the cognate protein that's mostly expressed in T cells, but there has been actually also very recently an abstract at ASH that has shown that ZAP70 also is maybe expressed in B cells. And so it may be that we're seeing these B cells, once we knock out sick, that we are still detecting active ZAP70. So that's under active investigation. But if we actually then look at our metabolic markers and divide these cells into 4 populations, the wild type that are phosphosick positive or negative, and the knockout that are phosphosick positive or negative, we can start to see some differences based on these different pathways. So here I'm showing lactate dehydrogenase versus GAPDH. And we can see that both in the knockout or the wild type, there is increase in these 2 proteins in the cells that have active SYK. And also we see just in the knockout without active SYK signaling, they still have higher levels, particularly of the lactate dehydrogenase. Then here showing some features of the TCA cycle or OXPHOS. Again, there's differences in the cells that are signaling as well as in the knockout cells. And again, we can start to see some differences with PAX5 and enolase 1 on the bottom set of plots, enolase 1 being the final enzyme in glycolysis. Sorry, I have a very touchy mouse today. Anyway, so we're really excited about integrating these sorts of metabolic markers into our sort of bigger B cell CyTOF panel so that we can start to put this all together and eventually apply to primary patient samples in order to come up with potentially a sort of a metabolic relationship with pre-BCR signaling cells. So to summarize this part and Yuxuan's work so far, SYK, we're thinking as a readout of pre-BCR activity in ALL cells, seems to be upstream of activated CREB NS6 and others, as we would expect. And that total SYK, or even possibly phospho-SYK, has a relationship to regulate PAX5 levels. And so we need to understand more about how does that regulate what PAX5 does transcriptionally. And then SYK and phospho-SYK show metabolic changes compared to wild type and or non-pre-BCR signaling cells. And we're really focusing on oxidative phosphorylation. Once again, Marcus Muchen's group has published in the past that PAX5 does seem to have some role as a metabolic gatekeeper in B cells and in ALL cells. So we're trying to understand how we can actually reconstruct this figure here to understand the relationship between pre-BCR signaling, PAX5 and PAX5 activity, and potentially metabolic reprogramming as a means for cellular resistance and potentially relapse. So now I'll transition into the second story I wanted to share as an extension of what we've been doing. And this is all work that's been led by Pablo Domizzi, who's a postdoc in the lab here, shown in Hawaii in pre-COVID days. In that Pablo's been looking at using our same tools to predict CD19 negative relapse after patients receive CAR T cell therapy for relapsed ALL. So as I showed this figure before, that CAR T cells, which are using engineered T cells to recognize CD19 protein as a means therapeutically to treat relapsed ALL, it's been tremendously exciting and successful. But we know that these therapies still are not helping us really get durable remission in the majority of patients. And so we wanted to take a look specifically at patients who relapse with CD19 loss, which in the Eliana trial was the majority of patients who relapsed, that they eventually downregulated CD19, and then the leukemia cell becomes essentially invisible to the CAR T cell. So taking the same approach of trying to understand what patients may be at risk of CD19 negative relapse and what are the cells that are responsible for that. There's already been a bit of work that's been done in the field to understand CD19 negative relapse. And so frameshift mutations in the extracellular domain have been described. We have seen transdifferentiation to a myeloid lineage, so cells that basically turn from looking like ALL to looking like AML. There's been disruption of 19 membrane trafficking, so 19 gets stuck inside the cell instead of making it all the way to the surface. And then Elena Sotillo, who's in Crystal's lab now, had described with colleagues alternative splicing of CD19, which enables resistance to CAR T cells. And then finally, a follow-up to that was intron retention of CD19. So we understand that there are potentially a lot of ways that this could happen genetically with CD19, but we were interested in taking a look at it on the protein level. So Pablo came up with two different hypotheses to potentially explain 19 negative relapse. One being that at the beginning, all of your cells have sort of uniform CD19 expression, and with the pressure of the CAR T cell, which is exerting pressure on 19, that the cells are able to in some way downregulate 19 and become CD19 negative. Another option could be that you sort of have these hidden CD19 negative cells that are already there before the patient gets CAR, and so that those cells are then capable of growing out and recapitulating the leukemia after the rest of the cells are taken care of by the CAR. So we had some initial preliminary data, and all of this is coming from patient-derived xenografts that we used in collaboration with David Barrett and Stephen Grupp at CHOP very generously. So we started out with 4 pairs of patients who had CD19 before they received their CAR and then had a CD19 negative relapse after. We did our very similar ALL panel by CyTOF, and here just showing in this MDS plot that we could then really see that these patients separated just by their phenotype, their kind of global phenotype, based on their mode of 19 loss. So this patient over here in the corner had the isoforms, whereas these 3 patients in the other corner had CD19 gene mutations. And of course, they separate away from the healthy control bone marrow. When we put these through our developmental classifier, we again saw, as expected, sort of the peak cell populations occupying the transition between pro-B cells and pre-B cells. But what was really interesting to us was we found this kind of enrichment here at the relapse, the 19 negative relapse. The classifier was calling these cells early non-B cells, which frankly, prior to this project was a population we didn't pay much attention to. It was sort of a population that we couldn't well identify because it didn't have a lot of particular B cell features, which is how the panel is designed. So again, with this collaboration, we've got a bunch more samples. So we now had over 40 samples for patients that both had complete remission after their CAR, patients who suffered CD19 negative relapse, patients who suffered CD19 positive relapse, and then some patients that were completely refractory to the CAR and had matched relapse samples or the post-CAR sample for many of these patients. Pablo took a look then, same approach with the CyTOF. When we look globally at these different groups, we could see that really there was no difference in either the percentage of CD19 positive cells pre-CAR or post-CAR, except in the case of when they lose 19. And then if you look in those CD19 positive cells, the median value of CD19, sort of a surrogate for the density of 19, also was not significantly different. So 19 alone was not something that seemed to be pointing toward any particular outcome. When we took that bigger cohort and did the developmental classifier, again, we saw this enrichment in our early non-B cell population. And so we got to thinking a little bit more about how our classifier works. And of course, it actually depends on CD19 as a way to classify the cells, because CD19 is such an important protein. We think about it as an important protein to identify early B cells. And so Pablo decided to give a try if we just excluded CD19 from the classifier and tested it first on our healthy controls. And in the healthy controls, it didn't really make a difference in the classification of cells if we used 19 or didn't to actually identify the cells. But in the leukemia cells, it made a difference in some cells. And so it actually revealed a population of cells that we called ProB2 discordant cells, meaning that if we use the classifier with CD19, we called them ProB2 cells. But if we excluded only CD19 from the classifier, they became called early non-B cells. And this was in contrast to some cells that it didn't matter if we had or did not have CD19 in the classification, it always called them ProB2 cells. So then we went back and said, okay, we have these discordant Pro-B cells. Do they only happen in CAR patients? And the answer is no. We had 22 samples in a collaboration with Charles Mulligan at St. Jude of diagnostic patient samples, never saw a CAR. And these patients, 17 of them who had more than 100 cells in the Pro-B2 population, also had these discordant Pro-B cells there. And so then if we looked across now, including these de novo ALL samples, but the additional ones who've been through CAR, we could see that we didn't see any discordant Pro-B cells in healthy bone marrow, but they were there in all conditions, all of these sort of clinical groups from the CAR cohorts. Pablo looked a little bit more deep into these cells. What are these Pro-B2 discordant cells? And this is comparing them between patients who went into a complete remission in green versus patients who had CD19 negative relapse. CD19 wasn't different, but our friend PAX5 appeared to be different. Phospho-NF-kappa B was different, and Icaros was different, although not as a univariate comparison statistically. But if we actually looked at them now, as a combination, so cells that are discordant Pro-B cells, they're of Icaros positive and PAX5 positive, then we start to see actually there are some differences in which the patients who are going to go into complete remission have higher levels of both of these transcription factors. So we started to think about the relationship between these transcription factors and CD19 regulation. CD19 is a known transcriptional target of PAX5. Icaros, less clear how it relates to CD19, but we could see that these discordant cells with low levels were prone to downregulate CD19 expression. Pablo then went and wanted to take a look at some of these samples using AbSeq, which is a version of antibody-based sequencing where you can do whole transcriptome and get surface proteome information through oligotagged antibodies. He looked at this in 3 of the pre-CAR samples who were going to go into complete remission, 3 of the patients before the CAR who would have a 19 negative relapse, and then 3 of the actual 19 negative relapse with a healthy control. Again, we used our healthy control as a way to project this single cell RNA sequencing data. So here's the healthy UMAP, 17 clusters of healthy cells. And if we put our ALL samples on this space, again, we can see that there was primarily an enrichment in 3 clusters. Cluster 6, which is primarily dead cells, we think. Cluster 14, which we think are early Pro-B cells, and then cluster 7, which I'm going to focus on, which we think look like pre-B2 cells based on their gene expression. So if we took cluster 7, and there was 353 differentially expressed genes between the patients who are going to have a complete response versus those that will have a CD19 negative relapse. And if we just do PCA based on those genes, we actually get a really beautiful separation between those 2 clinical outcomes. And we noticed that if you do a protein network enrichment, that there was, as we had seen in potentially in the CyTOF data, that the patients who are going to have a complete response have a more strong relationship between CD19 and Icaros, now at the transcriptional level. So we wanted to then ask, does Icaros influence CD19 if we downregulate Icaros either by shRNA knockdown or lenalidomide, which is a drug that is known to cause ubiquitination of Icaros, what happens to CD19? Pablo did this in 3 different ALL cell lines, differing on their BCR status, pre-BCR status, and also one of them has BCR-ABL translocation. And both with lenalidomide and the shRNA silencing, we saw that with increasing doses of Ikaros knockdown, we saw decreasing levels of CD19. So now we've taken our model where we believe that within that initial sample before the patient gets the CARs, there may be these hidden CD19 positive cells, but they're a little bit different when you look on the inside of them. So there are these discordant pro-B cells, and that patients who have higher levels of Ikaros and potentially PAX5 are the ones that are likely to go on and have a complete remission as long as their CAR continues to function, versus patients that may have lower levels of these transcription factors may be more prone to CD19 negative relapse. And Pablo is doing some further work both with manipulating Ikaros to try to understand the mechanisms behind this. And we're also trying to understand from the patient cohort that we looked at how many of these patients may have had Ikaros mutations, which are known to be a poor prognostic mutation in ALL. And so potentially enriched in our cohort of relapsed patients, but we're still tracking down that data. So just to sort of hopefully summarize the stories that I've shown today, we really are excited and enjoy using these single cell high dimensional studies, where we're looking at lots of different factors on individual cells in these cancer cells to provide insight into the most relevant cell populations for further study. There's a lot of challenges to doing this. And cancer heterogeneity, which was in the title of my talk, is really, a big one, is trying to understand with the huge diversity of data that comes from these different assays, what are the most important cells to look at. But in our hands, using sort of a healthy tissue as a way to line up your cancer cells has been very helpful and really given us a lot of insight. And so with that, I want to thank the folks in my lab. Since we haven't been able to have a full lab picture for the last almost year, we decided to do it by little avatars and Bitmojis, but I just feel very, very grateful to be working with such awesome, smart, wonderful people. And I only really touched on 2 projects, but there's lots of other great stuff happening, and I'm always happy to talk about that. And then, of course, collaborators and funders are so important, and we're very lucky to have wonderful collaborators and funders. And with that, I will stop.
Wow, that was really great, Kara. Thanks so much. Again, a tour de force of tumor resistance. For those that want to put additional questions in the chat and in the Q&A, please do that. I'll start off with a couple questions which came to mind. And so you talked about the difference between pre-existing resistance and induced resistance, both for the standard chemotherapy and for CAR T. But can you talk about whether there's, they seem to be different populations, whether it's CAR T or the standard, but they revolve around PAX5 and SIK. And so the question is, do you see the same SIK regulation in the CD19, or is that a parallel or different pathway?
Yeah. So I think that what we have seen in both of these stories, even though they're a little bit different, is that the sweet spot, I will say, of like developmental action that seems to be happening here is in this sort of transition between the pro-B to the pre-B cell population. So in both the CAR story and in the just the relapse in general story, that is the developmental space or time that the data points us to look at those cells. And in Pablo's data with the CAR cells, we definitely see that there's activation of the pre-B cell receptor. We see CREB is like almost always activated in a lot of these cells. And so it may be that there are, and again, these are patients that have already relapsed. So, we would expect that if both hypotheses hold true, that these cells would be sort of relying on that pathway, and whether or not that influences CD19 itself is a question, and I think, we'll have to further investigate that.
So then, I guess knowing it then again about Ikaros and PAX5 and SIK status will then help you predict both standard chemo and CAR T cell relapse, right?
Yeah, potentially.
And then I wanted to ask about the clusters of that you're seeing in resistance, because you talked about, like, you made it seem, at least, and you can correct me if I'm wrong, that there was like this single cluster of resistant population that you're seeing. And in other solid tumors, there's persister cells, which are non-dividing,
Mm-hmm.
that don't really differentiate, versus differentiating into other populations like T cell like cells or intermediate cells, which, I mean, PAX5 mutants do have can go into T cells in some ways, right? And so can you talk about really how many populations of resistant cells do you think there are in each of the different resistance populations you looked at?
Yeah, I mean, I think that a lot of this has to do with how you slice the bologna and
Right.
and how you cluster things. And in our in the original paper we published with the original 60 samples, we had them divided into like two populations, but they're developmentally contiguous, and that just was a function of like how we divided it. But it may be, of course, that that's not necessary. And as we sort of, I think, learn more with more samples and integrate additional assays and additional sort of information about these cell populations, it's hard to know if there's going to be if it's really one population or that has similar features that we just haven't fully fleshed out what all those features are, versus are there different populations in different patients. But sort of what our data is pointing to is maybe there is a somewhat of a conserved population across patients, at least for the ones that we've looked at so far.
Great. So there's a couple questions about the metabolomics you're looking at. Are there any biochemical data showing that changes in LDH or enolase affect metabolic activity, either in glycolysis or oxphos? And then I'll just add in there epigenetic status as well.
Yeah. So I think those are some of the questions that we're trying to, further flesh out, especially because, cellular metabolism, we're learning a lot, and, trying to go back to our basic biochemistry to remember and understand how these things are regulated. And definitely, I didn't even get into it, but a whole other part of Yuxuan's analysis has really highlighted differences in epigenetics, which we think Krebs may play in that, knowing what its role is. And that's always been sort of a suspect that keeps coming up in our analyses. So that's probably a whole nother part of that data that will get flushed out at some point when we have the hands, in the band. Yeah.
Sounds like an advertisement to come work in your lab.
Yeah.
And so do you think it's actually Krebs working directly epigenetically, or is it actually DNA methylation or histone? I mean, how do you work that out?
Tease it apart. I mean, my understanding with Krebs is it probably has to do with chromatin structure. So it's not, my guess is that it's not just Krebs directly and Krebs gene targets directly, is that, Krebs is potentially, the signaling of Krebs that we see is potentially a biomarker for some sort of epigenetic state or chromatin state that enables maybe these metabolic programs to be enabled. It may have to do with, actually, there's so many parts that are so interesting. It may have to do with localization in the bone marrow. Who are your neighbors from a stromal standpoint, and what's your environment with oxygen tension? All of those things are important. And there's been a lot of this has started to be fleshed out in AML. But sort of metabolic derangements as a mode of resistance in ALL is a little bit less, a road less walked.
Yeah. And then the obvious question, which is brought up also, can you tell us about small molecule Syk inhibitors in clinic? Do they mimic the metabolic effects that you see in vitro?
Yeah. So actually, Syk inhibitors have not come into ALL therapy yet. They are used in diffuse large B-cell lymphoma, and we've done some in vitro work with the Syk inhibitors and have seen with some of them that there is some downregulation of our signaling pathways. And I think we're probably going to have to go back to it a little bit more, but it definitely, because it was like one of those, we just tried this first and we didn't get real deep into it yet. But I think as we sort of flesh this out and understand a little bit more about how this signaling actually plays into what the cells are doing, Syk inhibitors may become something that really have a really important therapeutic role in ALL. And so being able to understand who may benefit, how do we know they're going to benefit, what's the biomarkers to trend that. But it's great that there are actual effective SYK inhibitors that are out there. I actually don't know how much they've been tested yet in children, but that's, something to look into.
And then other types of therapies that you've sort of mentioned, are there Ikaros inhibitors, or I know that there's some connection between Ikaros and STAT or JAK-STAT and TLR type analysis. I mean, have you thought about that area as well?
That part of it. I think we haven't gotten that far down as to like manipulate Ikaros. Ikaros also has been implicated in relapsed ALL, particularly that there's these dominant negative mutations, and Ikaros also is related to chromatin structure and state and is a really important sort of developmental transcription factor in B cells. And so trying to understand, is there a direct way to modulate Ikaros, or what is the output of having, a malfunctional Ikaros, and how could you target that? And the JAK-STAT story probably comes in also developmentally, because developmentally B cells that are before pre-B cell receptor signaling are focused on or are reliant on JAK-STAT signaling, typically through IL-7, and Ikaros does have a role in regulating that. So there's a lot of sort of interesting nuances to B cell biology or B cell developmental biology that I think probably play a role here.
Yeah, we have time for one more question. Let's see, none in the chat, so I'll just ask you, which is, clinically then, paint us a picture for treatment of childhood ALL coming forward, given the amazing work that is being done at Stanford, both in terms of therapy development for CAR-T and so 5 years from now, how do you see a patient being treated at Stanford based on the work you're doing?
Yeah. So I think that, there's a couple different ways. So one is in how do we do a better at prognosticating. So, one of the things we're really trying to do is bring that machine learning model, broaden it, learn more, get more samples, try to see, is that a way that we could actually bring that into some sort of a clinical diagnostic that you could actually even run in the clinical lab, not with a CyTOF, but with a flow cytometer, if we can figure out what are the most important proteins that we need to assay. And then the second part of it is, again, learning more about these relapse causing cells. Can we figure out other targets? And so in addition, like you're mentioning, could you target Ikaros? Could you target the signaling? That may be places that we're able to go more quickly to an immunotherapy or a cellular therapy, as opposed to waiting until patients are relapsed. And then also figuring out, are there modulators, small molecule modulators, that we could actually, improve the durability of those responses. So I think that, those are the directions that we're working in addition to trying to understand, are there other surface markers that might call out these cells? Yeah. Trying to do some sort of surface screening, because maybe they have a particular surfaceome that we could then exploit with a cellular therapy and as a way to find the cells.
Yeah. And then speaking specifically at Stanford, what's the greatest limitation of your work right now that might accelerate it quicker if it was available for you in your lab and your group?
Yeah, I mean, most of our work, actually, a lot of our work relies on primary patient samples. Our biobank locally is good, it's great, but we just don't have the volume of diagnoses, thankfully. So we are oftentimes having to go get samples from the Children's Oncology Group, St. Jude. We've had samples from overseas for collaborators in Italy, which I highly recommend. But it takes time and effort and a lot of paperwork to get those samples. So, we're trying to build that capacity here, trying to build a more rigorous sort of patient-derived xenograft, capacity here. So that's always a hurdle to get through. But, it just takes a little effort.
Great. Well, I just, on behalf of all of us, I just wanted to thank you for the great presentation, also the amazing amount of work you're doing, and for the patients as well as for science. And so keep up the great work. And also, just if anyone wants to talk to Kara more, please contact her. Please work in her lab. come to the lab and work and do this amazing stuff. So thanks so much for joining us. And we'll see you next month, March 1st, with Michelle Manja and Juliet Knowles. So thanks very much, Kara.
Thank you.